Okay, so many of you know who I am. I'm Dawn Field. I'm at the Center for Ecology and Hydrology. I also have affiliations and a joint appointment with the university. But there are lots of new people here today. So I actually founded the GSC with a bunch of other people way back in 2005. We've had continuous meetings. And I'm now the chair of the board, so we have a very active board. And as hopefully you'll start to pick up, the GSC is very much about active participation. If you have an idea or something you'd like to bring into this community, please do. Certainly this is how the Genomic Observatories network came in. And the reason there's so many new faces here is that there's a whole new sort of expansion of who might be working in the GSC. And it's these sites that have particularly good track records in long-term research who are now starting to champion omics and think if we could pull these sites together, what kind of research could be done. So I'm going to give a quick overview of the GSC, because the whole first session is a bit about what the GSC does, and then we'll go very much into the Genomic Observatories network. Right, so we're all facing this, I think, across all of our research activities. There's just too much biological data. This happens to be out of Darwin's notebook, so the first phylogenetic tree, perhaps that's sort of the birth of bioinformatics. Everybody here has data that you're somehow trying to put into computers, and this is sort of a whirlwind introduction. We all know about the explosion of data, we all know about the exponential curve, a lot of people are calling about the tidal wave, the tsunami of data. I'd rather think about it as a data bonanza. If we can get our act together and really have hands on the data and interpret it, they're huge opportunities. So here's one of these plots where you have the drop in sequencing cost and the accumulation of global data. So we have a lot of shared data at this point and more that could be fed in. What we really want to do is try to contextualize it. So looking to the future, we have to remind ourselves that they only figured out the double helix in 1953. There's several key inventions, then sequencing, PCR, so you can manipulate DNA and pretty much do what you wanted with it. Blast searching, the fact that you could do quick homology searching on computers really pushed forward the field of bioinformatics. The internet developed, that changed what everybody was doing. Suddenly you can get data everywhere, and we can actually imagine linking up data with other disciplines and beyond. Then we have the genomics revolution, from Homophilus to the human genome. I haven't continued that timeline, but it just increases in pace. We now have thousands of genomes, thousands of metagenomes, huge mega sequencing projects that we'll hear about. And you end up with this sort of map, which happens to be of sequencing capacity. And the main thing is that there's lots of sequencing capacity in the world, including BGI in China. This idea that you can have 4,000 people in one center doing genomics, omics, thinking about how this is going to revolutionize everything from the economy down is a completely different landscape. So what we're thinking about is, where is this going to go? Right, so the GSC, the Genomic Standards Consortium, a lot of us do science. I think most of us would not do standards for standards' sake. We simply want to do science better, and for a lot of this now, what's missing is context. So just a bit about it. We're open membership, and if we have a tagline, it's working towards better descriptions of our collections of genomes, metagenomes, and gene marker sets. As we all know, people are contributing in bits and pieces into this huge set. Somebody needs to think about sort of what the whole set looks like, and we have fantastic resources like NCBI, GenBank, EMBL. They're huge groups, MG-RAS. There are a lot of groups that are paving the way in this, but there's also a layer where we can think about this as a whole community. So we came together in 2005 to think about what extra would you want to describe about a genome sequence when you put it in the public domain. And it's hard to believe, but there was no next generation sequencing back there. At the first GSC meeting, we actually talked about what would you do with a 454 machine, if you even had one. It probably produces crap data and too much. We don't even know what to do with it, which is shocking. And when you looked at GenBank genomes, they weren't even putting strain names on, for example, strains from the same species, because they didn't expect that they would get duplicate strains. Now, of course, we know we've got pangenomes from lots of species. So metadata and context became more and more important. So from that first sort of exploratory meeting, everybody said we need to get together and make a step towards trying to get this kind of contextual data in. And we came up with a standard as a result of about 3 years' work. But we took the normal consensus path, and this applies to any projects that might come out of this community. Now again, this is a path that every community takes when you're trying to build something that only the group can build. So identifying the problem, define a community, the right balanced community, and hopefully we keep bringing people back into this meeting who can then form new communities, subgroups. Define the scope, implement a solution, and gain adoption. So some of our projects have now sort of gone this full circle. We'll talk about the mixed standard. Others are just getting started. Our mission, implement new genomic standards, figure out how we exchange data better, and in general, harmonize these activities so that we're not reinventing the wheel over and over again, that we can share some of this activity. And of course, what we spend most of our time doing is organizing meetings, but we really want them to have outcomes. So we're most interested in forming working groups and having these consensus products, whether they're standards or some of the other projects that we'll talk about. Okay, so I talked about this standard. The reason we formed was to come up with this minimum information about a genome sequence. It also covered metagenomes. And the key question was, we weren't collecting the who, where, when, what. We weren't getting latitude, longitude. We weren't getting anything beyond sort of the most basic stuff that went into GenBank, EMBL, DDBJ. And of course, this has evolved with the primary databases as we go, but we're talking about detail about the taxa, the habitat, being able to say these are soil sequences, these are water sequences. We're still getting closer and closer to being able to do that. Date and time, as we'll see, we'll talk quite a bit about Ocean Sampling Day. Having simultaneous sampling makes a lot of difference. It corrects for a lot of variability. Environmental measurements, pH, temperature, some of these things that we tend to think only environmental biologists are interested in now, but the human microbiome people are becoming as interested in this as we are. Details of DNA extraction methods, sequencing methods. And again, when we started, there was only Sanger sequencing. Nobody thought about multiple sequencing methods, and of course now we've got multiple generations. All right. So we've published 2 papers now, and this is only, it's not to go into detail, Palin will talk about this a bit more, but they are in the public domain, and you can submit this kind of data into GenBank, EMBL, DDBJ, and a variety of other databases. Right. So we are encouraging people now to do it. It still can be a bit cumbersome, but for those who are willing, we can provide help, and hopefully this will become easier as more projects start to implement. We also have a range of other projects. Again, I won't go into detail. We'll try to get this all up on the website. We've got an XML exchange format. We've got an identifiers mapping project across all the genomes so different databases can pull back the same data. We've got a journal that George Garrity will talk about. Microbial Earth project is a large coordinated project to sequence type strains. We have a developers group. There's something called M5. Volker won't talk about it too much, but sort of next generation infrastructure. There's a biodiversity genomics working group, and there are a variety of other projects now. We've just initiated having a project template form so that if new projects come up, we can sort of give a voice to these projects and make them public. We haven't redone the website and put them up, but they'll come online soon. So this meeting now is a chance to bring genomic observatories into this context. And many of you have been involved in sort of the thinking that's going on in this. This actually happened historically at GSC14, I think. Neil came to it, and we got talking about how Jack and collaborators and we had been doing work at L4 at Plymouth with microbial samples. And because you had 100 years of data, everything from contextual data in situ up to satellite data, you could really make an impact with microbial work. And Neil has Maria, he'll be one of the first genomic observatories to talk, and we started thinking about place-based science a lot more, and there are a lot of premier sites, and they've come together in this room. There are more that are engaged outside, but how do we start doing this DNA-centric place-based research? Because it's our best chance to do contextualized research, very highly contextualized research. So we had a short paper together. There were 68 authors on this. Not everybody's here, so the network already is sort of de facto formed. This meeting is about really deciding how we go forward, people getting to meet each other, not only the heads of sites, but the researchers using the sites, networks that include the sites, and of course the GSC, which has the genomics and the bioinformatics and the standards. Right, so I just wanted to say 1 or 2 words about standards. A lot of people think standards are the dry, boring end of things, and in many ways, like I said, most of us want to do science. We simply want to have enough consensus that we can build the tools that move the data around easy enough. And I just wanted to highlight the good things. It's supposed to be a convention. It's supposed to be a common language. It's supposed to lead to innovation. If we think about the internet, we all come in here and we all get on the internet. There are hundreds of standards in the technology stack that let you all just get on the internet and not worry about it anymore. That's the way that we're trying to think about standards here. And the other thing is that the principles are that not everything has to be standardized. None of us are here because we want to standardize things or squelch innovation or spend our time doing this. I would be rather sitting down with a grad student looking at all this great data and pulling patterns out of it. So if it doesn't need to be standardized, we're not going to do it. We still want innovation to come out. We need to pick the right concepts. And of course, it needs good groupthink. We need a good balanced thing and buy-in across the board. So we're not pushing standards per se. We're pushing sort of this idea about exploiting scientific data. Takes both technological and sociological changes. A lot of handshakes have to happen. How do we talk to the ecological data sets, the environmental data sets, beyond genomics? So that's, in a nutshell, what the GSC is supposed to be doing. We're up and running. We welcome the GOES, Biocode Commons, other projects that might form during this meeting. There's lots more information on the website. Just to say a big thank you to the board. We have a very active board in the GSC. And the funders, most of this is volunteer. And just to flag up that we are continuing meetings. There'll be another one in NCBI in April, and another one in Australia 6 months later, sort of on a rolling basis. So we're having calls for the next sets of meetings. So welcome to hopefully a very active participatory engagement. Thank you.